Okay, class, and last time in class we finished up the basics of the history of Mendel and how he developed all these concepts and a lot of terminology, right? Heterozygous, homozygous, dominant, recessive, etc. We also did your small monohybrid four-square Punnett squares. Now today we're going to look into our second learning objective for this chapter, which is us being able to calculate genetic probabilities based upon some traits and some given information. So you all pay attention here when we go over this, because I'm going to walk you through this one, and then we are going to do an example. Well, I'm going to have you guys help me fill in this example next. All right? So pay attention here. Let me show you how we do this. Okay, your alleles that you're going to be working with are going to be based upon your parents, right? So we have our F1 generation here. And now our F2 generation, you should remember from yesterday, is when we cross two individuals from the F1. So what we're going to be crossing is okay, there's our two parent allele combinations and their genotypes. But what we have to figure out, and what we haven't learned yet in these dihybrid crosses, is how to figure out the individual allele combinations for each parent. I'm going to show you how to do that. Okay, it's really simple. First thing you need to do is figure out what individual alleles this dominant Y could interact with. I write out these arrows every time when I'm doing this just to make sure I get it right. That big Y can combine with the big R, and we could have that as one of our allele combinations. And we see that right here. So you'd fill that in as one of your allele combinations. We have to figure out our second one. This big Y could also combine with that recessive lowercase r. So let's go ahead and write that down. Now we need to go the other way and look at this second Y and its combinations. And the process is going to remain the same. We'll just draw that arrow there. Make sure you're careful with your letters so you can recognize the little y's from the uppercase ones. It can be tricky when it's the same, when the uppercase and lowercase are the same. And then finally, that combination there, little y, little r. Now, since both of these parents are the same, we know that this parent over here is going to have these same allele combinations. So we don't need to do that again for this one. So if you look here, big Y, big R, big Y, little r, little y, big R, two lowercase recessives, little y, little r. Raise your hand if you all understand how we calculated and figured out all of those allele combinations based upon the parents' genotypes. Aiden, I don't see your hand raised. What don't you understand about this?
So do you always put like the uppercase dominant allele first?
Okay, that's a common question. If you all couldn't hear, what Aiden asked was, do you always put the uppercase dominant allele first when you're writing these sequences of the individual alleles? The answer is yes, you do. Geneticists customarily are going to put the dominant allele first. However, if you don't, you're going to get the same answers, but it is expected that once you learn how to do this, you put the dominant allele first. It's a good question, Aiden. So after we figure out all of our allele combinations, we go ahead and fill them in. Now, since here both parents have the same genotype, you don't have to worry as much about marking it as male and female. But you see here how we have the axes marked for the male and for the female. Okay, so we fill those in, and our y-axis here on our chart is going to be the same, right? Because both of our parents are the same. So now the next step is for us to begin filling in our squares. Now to me, this is the fun part, because you got all the setup done, and now you just get to plug in all of your letters. And I'm sure looking at this, you guys kind of can see where we're going here. Let me fill in this first line here for you. Big Y, big Y, big R, big R. So all you do, you just go across and then down for each one of them. So you can see here how we end up with two big Ys, two big Rs, Right here, how we end up with two big Y's, one big R, one little r. And then here, one big Y, one little y, two big R's. And then finally, one big Y, one little y, one big R, little r, get you a heterozygote. Let me just fill in this bottom one to make sure we got it down. Two big Y's, big R, little r. Okay? Do you guys understand? Raise your hand if so. If we all understand how we're going to fill in these boxes here. Oh, great. We got a bunch of geneticists out here in the audience. Fantastic. All right, guys, if you all understand that, let me go ahead and clear this off. And let's do our next step, which is after we get all these filled in, we then have to calculate our phenotypic ratio. Now, typically, this is what I'll be grading. Okay, I'll give you partial credit when we're looking at these problems on our homework and stuff, but this phenotypic ratio is basically your final answer that we'll be looking for. All you need to do is look at the individual genotypes that show you each individual ratio here. So here we have yellow and smooth. So that's going to be one big Y, one big R. Remember, guys, with the dominant alleles, you only need one of them. So anything that's yellow and smooth will look like that. Have at least one big Y and one big R. I label these when I'm doing these with little symbols. We'll make that one a star. So every time you see one big Y and one big R, write a star in there. Now this is easy, the way I have this example set up for you, because you got pictures of what the seeds actually look like, right? You don't just have genotypes there. You actually can see that it's yellow and round, that it's green and wrinkled. That being said, you're not going to have that when you all do your homework and stuff, right? You're just going to have a bunch of genotypes in there. So it's going to be more important that you label these accordingly with each individual label. Let me do one more for you, then we'll move on to our example. For example, let's do yellow and wrinkled. So yellow's got to have one big Y. Wrinkled, two recessives we have to have. So then let's go find those. Let's label those maybe with a circle. So here, at least one big Y and two little R's, right there, there, and there. So we have three of those. Okay, so you just count them up for each individual one. You can see here we'll end up with 9 yellow and round, 3 yellow and wrinkled, and then to finish it off, we have green and round. That would give us 1, 2, 3, and then green and wrinkled would give us 1. All right, so that's how we figure out our phenotypic ratio, approximately 9 to 3 to 3 to 1. Okay, so it seems like you all got that down pretty good. Let's go ahead and look at an example and do this one together. Let's start to fill this one out, and then you all can finish it for homework for tomorrow. Okay, we have another example here, just like the one we just did. Okay, the process is the same. In this case, we're looking at what's called a cyclops. And a cyclops is a type of microscopic freshwater pond animal. They're relatively common. And we have a couple traits up here that we want to look at with it. Okay, we're going to look at the morphology of its antenna. And then we're going to look at whether it's resistant to pesticides or not. And then fill in our genotypes. So the first thing we need to do is fill in our genotypes. Those genotypes are going to be, you guys are going to figure this out here in a minute, so be thinking about this. Resistant to pesticides and smooth antenna, and then one that's heterozygous for both. So why don't you all get into you all are sitting together today, into your think, pair, share groups. And why don't you think about this for around 45 seconds or so, Aiden, if you want to get in with another group, go right ahead and chat with them for the next 45 seconds or so. And I'm going to ask you to help me figure out these parental genotypes for what's resistant and smooth and what the genotype will be that's heterozygote for both. All right? Anyone who has that figured out already can begin to think about our allele combinations that we're going to look at. Okay? So you guys take about 45 seconds and discuss this with your partners there at your desk. I'll walk around if anyone has any questions, and then we'll begin to solve this problem together, okay? Remember how we did that last one, guys, with the peas, with the round and wrinkle and green and yellow. Same process here. If anyone has it figured out already, why don't you raise your hand and let me know, and we'll go ahead and fill it in. Reagan, what do you think the genotype's going to be for the Cyclops that's resistant and smooth?
Little b, and little n, and little.
Okay. Reagan said all recessive traits, 2 little b's and 2 little n's. Christian, Jace, does that make sense to you? Do you think she got that one right?
Yes, I believe she.
I think so too. Good job. Yes, resistance is a recessive trait, so we have to have both little n's. And the barbs, smooth barbs is a dominant. Smooth antenna is a recessive trait, so we have to have both recessive alleles. Very good job, Reagan. All right, who wants to fill in the next one to help me out with the traits that are heterozygous for both? Aiden, what do you think the genotype for that parent is?
I have big B, little b, big N, little n.
Okay. Big B, little b, big N, little m. Yep, you guys didn't need to do much for that one. The fact that it said heterozygous for both, we know what heterozygous means. So we're doing great towards accomplishing our second learning objective for this chapter, which is for us to be able to calculate all these genetic probabilities and do some basic Punnett square analysis. So it looks like you guys are progressing nicely to this. Now, remember our next step after we did this in our last example. Our next step was to figure out our allele combinations. You guys stay in your groups, give you about another 45 seconds or so. Remember how we figure out our allele combinations for each parent. Well, these ones are going to be different, right? These two are the same. These two are going to be different. So let's figure out our allele combinations. I'll give you guys about 45 seconds, and then I will check in with you and see how we're doing with figuring out our allele combinations.
For the top, B. We have B, little b, little n, and little b.
I hear it sounds like some of you got this figured out already. Who thinks they got some of our genotypes filled out? Let's do these resistant and smooth ones first. I think those are going to be pretty easy if we know what we're doing here. Who wants to help me fill out some of the genotypes for the resistant and smooth one? Jace? For resistant and smooth, what's one of the allele combinations that we're going to have, right? Remember how we do it with our moving of the arrows there.
Little b, little n.
There you go. Little b, little n. Now, what's the rest of them going to be?
The same.
Yeah, there you go. We're all figuring this out. They're all going to be little b, little n. So let's go ahead. We can fill all those in. You guys fill this out on your worksheet. All right. Now, the heterozygous for both, that's going to be a little bit more tricky. Who can help me figure out some of the allele combinations for this one here? Hayden, what do you got? Give me one of them.
Big B, big N.
Big B, big N. So what Hayden just did was he did his arrow like this, his first arrow. And he said big B, big N, 2 dominant alleles. Good job, Hayden. Go ahead. What do we got for another one? Yeah. Big B, little N. So you guys are getting this down. Big B, little N. We got 2 more. Hayden, give me one more.
Little B, big N.
Yep. Now we do these ones. Little B, big N. Last one. Jace, go ahead. There we go. Little B, little N. Very good. All right. Final step here. Almost final step. Our next step, just like we did in our last example, we now have to fill in the boxes. Why don't you guys take about 30, 45 seconds or so. We have a few groups here. Hayden, your group, you and Reagan, why don't you work on line 1 for us? Jace, Christian, and Aiden, why don't you guys work on line 2? Isaac and Connor, why don't you guys try and work on line 3 for me here? So take about 45 seconds or so. Then we'll check in on that. We should be able to fill this table in.
It's big B, little N, so it would go big B, little B, and then little N, all the way. And then everything else would be the same.
Yeah, here, okay. Big B, just a little B, big N, just a little N,
Remember how you do it, guys. You keep your letters together. Put your dominant allele first. And then you just basically go across and then down to fill in all of your squares. Seems like some of you are kind of figuring this out already. I think we're getting pretty good at this. Group number 1 here, Reagan and Hayden. What do you guys think we got here for our first box here?
they'll all be the same, so big B, little B, big N, little.
See, Reagan realized something there. She realized that all of these are the same allele combination. So I asked her what the first box should be, and she told me that they're all going to be the same, and that they're all going to be big B, little b, big N, little n. And then the rest of these will all fill in the same. Christian, what about your group? Your group number 2 here to figure out this second line. What do you think our first box is going to be here in our second one?
So we got big B, little B, and then little N.
There we go. Big B, little b, little n, little n. And once again, all those will just fill in for the remainder of the boxes. Connor and Isaac, why don't you guys help me out with this third one? Go ahead. Yep. So here we got little b, and then we got big N, little n. Once again, those will continue on. Finally, I'll do the last one for you. You guys got this figured all out. 2 little b's, 2 little n's. All right, and that will fill in all those. So your last step, you guys can complete this and have it for tomorrow. Calculate your phenotypic ratios. So look up each one of these genotypes and what the associated phenotype would be. Right, so big B and big N, that's going to lead to barbed and non-resistant. You all finish up with the rest of those, and I'll check those off tomorrow when you all come in. Does anyone have any questions based upon us in our calculations that we just did? Jace?
Are there any examples when alleles do not exhibit complete dominance?
Oh, that's a good question. Jace asked, are there any examples where alleles do not exhibit complete dominance? And that's a good question because that kind of leads into some material that we're going to be looking at coming up more on next week. But since Jace asked that question now, let's go ahead and look at this example now. And what you're looking at here answers your question, Jace. Not always do we see alleles exhibit complete dominance, right? In this case, we see an example of what we call incomplete dominance. And this is in a snapdragon flower. So here, okay, pay attention, guys. We haven't learned this yet, and it's a good question, Jace, because you led right into something that I was going to be talking about. Here you have what's called a red snapdragon flower. We have them around here in Ohio. They're a local flower species. And then here you have a white species. Now notice your genotypes. C stands for color, and then the superscript represents the actual color of that individual. So R for red and W for white. We can see our gametes here are both going to be CR and CW, so that's where these come from. Our F1s are all going to be heterozygotes. Now we're going to do our F2s. The F2s are just CR-CW times another one, CR-CW. Okay, so we got those in here. Now we're even back to our little 4 square Punnett squares. We're not even doing the big 16 square one here. All right, so we'll go ahead and fill these in, both the same for the male and the female. And then we go ahead and just fill them in like we usually do. CR, both reds, get you a red flower. CW, get you a white flower. But we're seeing something weird here. We haven't seen this before yet. In incomplete dominance, the phenotype of the heterozygote is in between that of the dominant and the recessive phenotype. So in this case, our dominant allele combination is red. Our recessive allele combination is white. When you mix red and white, you end up with your pink flowers. Okay? We're going to discuss some more of these advanced concepts, most likely tomorrow. I gave you all a handout on epistasis, so we will pick up tomorrow. I'll explain this example on epistasis, and then we will do an example just like we did today. We'll do another example. So some of you, if you want to get ahead, you can kind of work on this problem for tomorrow. And then tomorrow in class, we can work through it together just like we did today. So is there anyone, raise your hand if you understand the incomplete dominance example that we just covered. All right, you all raised your hand. Great work. Good job today, guys. We'll pick up tomorrow with our third learning objective for this chapter, which is going to be understanding the concepts behind some of the more advanced concepts in genetics, such as incomplete dominance, epistasis, and we'll also talk about polygenic inheritance. Okay, so finish up that homework problem where you all finish up your phenotypes for this, and we'll pick up with the advanced genetics concepts tomorrow. That's all we got for today. Good job.